I'm just going to, perfect. So hello, everyone. We have with us, okay, I'm sorry, I'm letting people in while I'm, Maureen, do you mind doing that? It's popping up. Perfect. So hello, everyone. We have with us today Dr. Christina Ferrone, who's a recent PhD from Pathology and Molecular Medicine. Just a small introduction to Dr. Ferrone. She completed her bachelor's at Queen's in Life Sciences and did her fourth-year thesis project on breast cancer with Peter Creer here, and completed her PhD, MSc, and converted to PhD in Dr. Michael Ross' lab in April this year, and currently finishing up some manuscripts for submission. She has already published some of her PhD research work, as some of you might know. During her PhD, she has received a lot of awards and scholarships, and I'm going to highlight just a few here, the CIHR Banting and Best Canada Graduate Scholarship being the most important one, the Weaver Research Excellence Award for her publication, Dr. Robert John Wilson Graduate Fellowship, and several others. And she has in fact turned down OGS a couple of times. So throughout her grad studies, Christina has actively participated in several committees in pathology of Department of Pathology and Molecular Medicine, Faculty of Health Sciences, and several others. She also served as grad student rep, and recently I have watched her help coordinating Cancer 499 thesis course. She has also participated in a variety of mentorship programs such as WISE. Christina, maybe you could tell us more about what WISE is, and Let's Talk Science, a research mentorship program started by Dr. Elliott, who's here with us today. I'm very excited to see you, Bruce, here in collaboration with 4 Directions. So besides doing science, Christina loves or enjoys swimming and spending time outdoors, gardening in summer, and some knitting in winter. So she has a whole bunch of different interests. She has also worked at the Queen's Ark Pool as a head lifeguard or advanced instructor, training staff, etc. in later years of her degree. So today, Christina is going to share some of her PhD work focusing on the clinical utility of nucleic acid sequencing and myeloid malignancies and precursor lesions. And after her, I was just talking to her what she's going to do after her PhD and where she's heading. I'm not going to again steal the thunder. I'll let her tell if she wants to, or maybe we all can wait for a couple of months to hear that excitement. But Christina, take it away.
Thank you. Thank you for the introduction. In terms of WISE, your question, that's Women in Science and Engineering. So just mentoring students. It's been sort of an undergrad student per year kind of thing. That's in science.
I didn't hear about that before.
Yeah, no, it's okay. In terms of next steps, I was saying I didn't want to jinx anything because I don't have the sort of formal offer, but I have a potential position lined up to go to the National Cancer Institute at NIH in the Laboratory of Pathology there to do some NGS variant review. So sort of in line with what I'll be talking about today. Thank you. Okay, let me get this. So good. Yeah, so thank you for the introduction. And so as Dr. Cody mentioned, I'll be discussing the clinical utility and impact of NGS in myeloid malignancies and precursor lesions. And so just a little bit of general overview. I'm going to give a little bit of background on myeloid malignancies for those that are less familiar, and then go into a bit more on the clinical utility of NGS in these cancers, as well as some background on what we call myeloid precursor lesions or pre-malignant states. So a bit more about that, and then into the clinical utility and importance of incorporating that for precursor lesions, which has sort of been the focus of the later part of my PhD. So part that we're working on getting published. Okay, let's see about that. Perfect. Okay. Oh, let me click. Okay, so just a little background in general. So hematopoiesis is the production of blood cells, which involves both myeloid and lymphoid progenitor cells, which then give rise to their respective lineages of mature cells. Myeloid malignancies, however, occur when hematopoietic stem or progenitor cell mutant clones proliferate and expand and then skew blood production toward myeloid lineages, as you can see on the left here. Just going to adjust this. Perfect. Okay. So just in general, myeloid malignancies are characterized by uncontrolled proliferation and or blocks of differentiation of abnormal myeloid progenitor cells or hematopoietic stem or progenitor cells. So I'm going to start on the far right here. Actually, this is sort of an overview of the main categories of myeloid malignancies, and AML or acute myeloid leukemia tends to be one that people are most familiar with. So AML is characterized by the excessive proliferation of those immature myeloid cells, so these myeloid progenitors, whereas myeloproliferative neoplasms, or MPN, are characterized by the excessive proliferation of those mature myeloid cells. So we're looking at the downstream of this pathway, getting proliferation of those cells there. And an example that many people are familiar with as well is CML, or chronic myeloid leukemia, as sort of a main category of myeloproliferative neoplasms. Now, MDS, which is going to be a bit more of the focus of my talk today, or myelodysplastic syndromes, are actually characterized by what's called ineffective hematopoiesis. So the progenitor cells are essentially failing to properly mature. So we're saying bone marrow failure, which then in turn results in cytopenias or low blood cell counts in the systemic or peripheral cells. And then MDS/MPN is just sort of another category. So these are characterized by overlapping features between MDS and MPN. So I've touched just a little bit on the proliferation side of things, but another key feature of MDS in comparison to the other categories is that there's usually dysplasia or abnormalities in the cells in one or more of those myeloid lineages. MPN tends to be mostly normal. MDS/MPN, having that overlap, tends to have some dysplasia typically, and AML may or may not exhibit dysplasia. And so diagnoses of these malignancies involve assessment of patients' peripheral blood and bone marrow that's typically investigated through cellular morphology, cytochemistry, and genetic assessment. So I'm going to focus especially on MDS because it tends to be one of the more challenging ones to diagnose. And so typically MDS is diagnosed based on the presence of dysplasia, as I mentioned, in one or more myeloid lineages, and the presence of an abnormal karyotype if present. Now, the reason why this can be especially challenging to diagnose is based on the fact that MDS can be, dysplasia can be subjective up to interpretation. It can be mimicked by reversible causes such as alcohol, medications, nutritional deficiencies that can sort of look like the same dysplastic picture that you might see in MDS. So you need to be sure that those are being ruled out. In addition, abnormal karyotypes that are part of the diagnostic process are only actually present in about 50% of cases. So when that's not present, it does make it more challenging to confirm that MDS is actually the result of this dysplasia. And this is especially important given the prevalence of MDS in the aging population and how our population is especially aging at this time. So based on that, more accurate and precise diagnostic tools are necessary for early stage myeloid malignancies, especially MDS. But thankfully, molecular profiling through next generation sequencing has permitted the identification of many genes that are recurrently mutated in myeloid malignancies, especially including MDS, and especially over the last 10 or so years with the advent of this technology. So on the bright side, the WHO classification that was revised in 2017 has started to incorporate some of these recurrent mutations into the diagnostic criteria for myeloid malignancies, especially in MPN and AML. There's a few in there, but I want to sort of give an example of how molecular profiling can actually help diagnose myeloid malignancies, especially MDS, for example. So ring sideroblasts are an example of a dysplastic feature that can be seen in myelodysplastic syndromes. And so prior to the 2017 revision, the classification for MDS with ring sideroblast was referred to as refractory anemia with ring sideroblasts. And so ring sideroblasts is actually these sort of blue ring features around the nuclei of developing erythrocytes. And so what the previous diagnostic criteria required a minimum of 15% ring sideroblasts to reach a diagnosis of MDS. However, in 2017, the new category of myelodysplastic syndromes with ring sideroblasts actually only requires a 5% ring sideroblast amount in an individual to reach that MDS diagnosis if it is accompanied by a mutation in the SF3B1 gene. And so in terms of the mechanism, for those that are interested in terms of this connection as to why we know that mutation in SF3B1 and ring sideroblasts are associated, so normal SF3B1 functions as a splicing factor, and one of its targets is the ABCB7 transporter. And so this is a transporter that's responsible for transporting iron out of the mitochondria of developing erythrocytes so that they can be used for heme biosynthesis. However, when SF3B1 is mutated, we get abnormal splicing of the genes for this transporter, which can result in abnormal iron transport. And so this can cause accumulation of iron within the mitochondria and reduced heme synthesis because we're not getting that transport out of the mitochondria. What that translates to into dysplastic features is this ring structure here. So the iron is staining blue when the cells are being stained. And so what this is the mitochondria that are surrounding the nuclei of these developing erythrocytes filled with iron. And so essentially, the presence of this SF3B1 mutation helps confirm that the ring sideroblasts are not a dysplastic feature associated with some other reversible cause, but helps confirm that, yes, in fact, this is associated with the myelodysplastic syndrome, which allows patients to be diagnosed earlier on at a lower level of dysplastic features, potentially be treated earlier. Either way, earlier detection is always favorable when possible. So this is a good start, but what about the rest of the molecular features that have been identified and deemed relevant in MDS and other myeloid malignancies? So this is just sort of a general representation. It's not an exhaustive list of some of the genes that have been shown to be recurrently mutated in some of these diseases. So you can see that there's quite a bit of overlap between all of them. But there's also some that are more specific to the individual myeloid malignancy categories. And then AML especially tends to be associated with a good number of RNA fusions as well. BCR-ABL1, for example, as a one that people are often familiar with. Okay, oops, sorry. So these genes have been shown to have an impact on diagnostics, as I just showed for MDS, but especially in MPN and AML. And some of these have standalone tests that are part of the routine workup for patients that are suspicious for MPN or AML. Some of them have been shown to impact prognosis, and then some even have options for treatment in terms of targeted therapies. So based on all of this information, the main hypothesis for my PhD work was that initially my master's work, but of course we built on that as happens, but I'll get to the later part. Anyhow, targeted next generation sequencing in myeloid malignancies could lead to clinical actions such as informing diagnoses, clinical risk and follow-up requirements, the presence of druggable targets, and categorizing patients to select appropriate treatment options. So sort of some of the main objectives that we were looking at were first to validate the Oncomine myeloid targeted NGS panel. So this is a ion torrent sequencing panel offered by Thermo Fisher, targeting genes recurrently mutated in myeloid malignancies. Then we wanted to demonstrate the clinical actionability of impact in NGS in myeloid malignancies, as I previously mentioned. And then finally, the latter half, I'm going to focus mainly on these two objectives for today's talk. And the latter half, I'll be looking at the clinical utility of NGS in early stage MDS and myeloid precursor lesions, which I'll provide a bit more background on when we get to that part. So first thing, I'm briefly just going to touch on the validation of the panel. And so we validated, like I said, the Oncomine myeloid panel, which is an ion semiconductor sequencing assay that targets DNA and RNA. Targeted sequencing can be especially useful clinically because we end up with a greater coverage of specific genes that are recurrently mutated, especially in myeloid malignancies. Consider there's sort of such a set list of genes that tend to be recurrently mutated. And we were granted early on, early access to a non-commercial version of the panel while they were still getting it ready for its commercial release. So we were able to actually start that validation a little earlier too. And you can just see here the genes that are covered by the panel. So there's 40 DNA genes covered, including 17 full genes and 23 hotspot regions of the genes on the left. And then you can see the RNA fusion driver genes that are covered. There's 29 of them with approximately 700 or so possible fusion pairs that can be detected with that. But my focus today is going to be on the DNA side of things. So in terms of how we validated this, it was based on IQMH and ACMG validation recommendations, including sensitivity, specificity, test precision, accuracy, linearity, and limit of detection. So as I mentioned, I'm not going to go into too much detail about the actual validation today, but if you're interested, that was sort of the first half of my recent publication in 2021. And then the second half is looking at the clinical utility and impact, as I mentioned. So I'm going to go into that a little bit more. Okay, so in fall of 2017, which is when I was starting my master's, we started out sequencing individual cases with one-off consent from hospitals around Ontario. So people were contacting us to have their patient samples sequenced so that it could be used clinically to help inform their care. As of April 2018, however, so only 8 months later, Cancer Care Ontario offered funding to sequence samples for patients with AML. So that was very helpful because now there's funding, we can sequence those with AML, but only those with AML. And so because KHSC is a smaller institution compared with some of the larger ones, we didn't actually have enough AML cases, thankfully, to fill up the sequencing chips as often as they needed to be sequenced, given the importance of a quick turnaround for these patients that have a very acute disease. So we were able to actually fill in the gaps on some of those sequencing runs to not waste the reagents with some other myeloid malignancies, such as MDS or MPN, and a couple other hematologic malignancies as well. And so we were able to use those sequencing results to be able to sort of look at the clinical actionability and impact beyond AML to hopefully demonstrate why they should also be approved for funded testing. So initially, when we were looking at the variants that we were seeing, we were determining their potential clinical significance based on this tier system that was proposed by Lee et al. in JMD in 2017. And so mainly the variants are falling into those first 3 tiers. So variants of strong clinical significance, potential clinical significance, and unknown. But we were finding that this was a little bit too rigid, and we were ending up with a lot of variants in that tier 2 section. Especially considering how, like I said, there's such a set list of important genes in myeloid malignancies. So we actually sort of adjusted our definition for the purposes of our research, and we qualified actionability or classified actionability based on the variant's ability to achieve, clarify, or support a diagnosis, their ability to inform prognosis, and their ability to inform clinical management. So in terms of management, that could be things like follow-up frequency, treatment options. And so I prepared sort of a large table. I can't show you, it's a little bit big for PowerPoint, but going through all of the individual myeloid malignancy categories and then each diagnosis, prognosis management for each gene, and highlighting whether it was poor prognostic, more favorable prognosis, whether it helped with the diagnosis, whether there were treatment options, whatnot. So we were actually using that in terms of classifying actionability. So this cohort was a prospective cohort that we were building between about fall 2017 to September 2019. So about 2 years worth of patient sequencing. So this included 187 samples from 168 unique patients. So some of them had repeat sequencing throughout the course of their disease. About 2/3 were male and 1/3 female, with an average age around 63 years, consistent with these cancers being associated with older age. So in terms of the distribution of the malignancies that we sequenced, we had about a quarter of those being AML, which are the ones that were, funded for filling up the chip. We had a good chunk that were suspected or known to be MDS, so suspected as in not quite meeting the diagnostic criteria, but being explored for potential molecular features. Some MPN, suspected or known, MDS/MPN, and then a few other hematological disorders. So there's a few BALLs, aplastic anemias, among others. So in terms of the actual process for sequencing these patient samples, individual patient samples were collected either as peripheral blood or bone marrow, and the DNA was extracted, and DNA library preparations were done manually early on, and then we shifted to the Ion Chef system later on once it was approved for clinical testing. And so that just sort of increases automation and reduces some of the human error in the process. And then template preparation was completed on the Ion Chef to prepare for sequencing. And as I mentioned, it was done on Ion Torrent NGS. It was Ion Torrent NGS using the Ion S5 sequencer. And then once the sequencing was completed, the results are uploaded to Torrent Suite and then to Ion Reporter. And in Ion Reporter, we're able to sort of go through the individual variants that are called, and then identify whether or not they might have clinical utility, for example, looking at databases such as COSMIC or the Catalog of Somatic Mutations in Cancer, ClinVar, looking at Nomad, for example, to see whether specific variants might qualify as SNPs if they're in that 50% variant allele frequency level. So just sort of the clinical interpretation of clinical actionability. So in this cohort of 168 patients, we had 299 variants that were detected. And out of those, 77% of them, so over 3/4, were qualified as clinically actionable based on our criteria. That could include even supporting a diagnosis that was previously identified. And then about a quarter were considered non-actionable. From that 77%, about half of them had an impact on diagnosis, about a third having an potential impact on prognosis, and then about a quarter impacting management. And I apologize for this Venn diagram. It's not perfectly to scale, but you can see that there's a good chunk that had overlap, so features that could impact all of these or only a few of them as well. What we saw in terms of diagnosis was that NGS was especially helpful in reaching diagnosis of query MDS. So you can see here, this is before NGS, and then this is after. So we had a good chunk that were MDS, but then a bunch that were, the question mark refers to query MDS, so subdiagnostic. And you can see here, some of them have an MDS diagnosis following sequencing. Others have some of these precursor lesions, which I'll touch on a little bit. Some with other, and then some that U is unclassified, unknown, still undiagnosed, so still unconfirmed potentially. And then also helpful for query MPN. And so you can see not so helpful on the diagnosis side with AML, because AML is a bit more clear cut to diagnose than MDS off the bat. And so then in terms of prognosis, I'm sure a lot of you are familiar with the idea that some of these molecular features could be prognostic indicators. But in terms of management, I thought I would share just a couple of examples of targeted therapies that could be available based on the presence of variants in certain genes. So midostaurin is a tyrosine kinase inhibitor that specifically can be used in individuals with AML with FLT3 mutations. Enasidenib is a newer one that can be used for those with IDH2 mutations, especially in MPN, but can be used in others that have IDH2 mutations as well, potentially. And then ruxolitinib, which is a targeted therapy for patients with MPN with JAK2 mutations. And JAK2 is a very prominent mutation that's seen in MPN, so that's an important one. JAK2 is one of the ones that standalone assays are done to confirm prior to or separate from NGS and whatnot. And so currently, though, there's more and more of these starting to be developed. So luspatercept is a newer one that's been shown to be helpful in patients with lower risk myelodysplastic syndromes, such as patients with SF3B1 mutations, because SF3B1 is associated with a more favorable prognosis and thus sort of qualifying in that lower risk category. And then also recently, interestingly, in the last couple of years, this APR246, which has been recently named eprinetapop, I'm not sure how to say that exactly, can be used along with azacitidine in MDS and in AML for individuals with TP53 mutated MDS and AML. So very interesting as well. So important to be able to detect these if treatments are available. So in terms of sort of clinical impact beyond individual types of variants that are identified, we saw that the presence of even a single variant versus 0 was independently predictive of overall survival. So that was independent of age, sex, or diagnosis within our cohort across the spectrum of myeloid malignancies. So even, as you can see, even having one is associated with a poorer prognosis, regardless of what that variant might be overall. So that really highlights the importance of detecting these and the use of NGS in myeloid malignancies. And so overall, we saw that NGS was especially helpful in cases of early stage MDS, as I mentioned, and identifying precursor lesions. So as you can see here, we had 71 patients that were suspected but subthreshold for MDS, MPN, or MDS/MPN diagnosis. And we saw 51% of these individuals had clonality detected, so at least one variant was present. So in some of those cases, that helps confirm the diagnosis of a myeloid malignancy or indicates the presence of one of these precursor lesions. And then another half had no variants, which in some cases prompted management of potentially reversible causes of cytopenias, or perhaps just informs follow-up frequency. Perhaps they don't need to be followed quite as closely as individuals that would have variants present. So that just really highlights the importance of being able to detect that clonality that wouldn't be detected otherwise for these individuals. So as I mentioned, that's going to bring me into sort of the myeloid precursor lesions in early MDS side. So I'm going to give you a little bit of background on what I mean by myeloid precursor lesions here. So I mentioned that MDS is diagnosed based on dysplasia and karyotype abnormalities, but initially it's typically explored due to cytopenias that then, prompt exploration for potential dysplasia or abnormal karyotypes. But what if we don't have dysplasia, or the dysplasia is subthreshold for WHO diagnostic criteria and the karyotype is normal? So for this state, we've got the term ICUS, or idiopathic cytopenia of undetermined significance. So this term was coined in 2007 to really describe individuals that have these cytopenias with an undetermined cause, but sort of not meeting that diagnostic criteria for myeloid malignancy. And so, as I mentioned, lots of variants have been identified, but what about cases where patients have no or subthreshold dysplasia? They don't have cytopenias like these ICUS individuals. Their karyotype is normal, but they have clonality identified based on NGS, since that has been developed. So for that, we have the term clonal hematopoiesis of indeterminate potential, or CHIP. So this term was adopted around 2014, 2015. It's also sometimes referred to as ARCH, or age-related clonal hematopoiesis. And so this term was adopted to consider those, again, that lack dysplasia, karyotype abnormalities, or cytopenias, but are exhibiting some form of clonality, so these molecular features through NGS. And so the reason that this is also called ARCH is because the mutations that occur in our hematopoietic stem cells or myeloid progenitor cells occur can increase over time as we age. So we can accumulate additional mutations, and the accumulation of mutations can result in disease progression where clonal advantage is conferred. And so, for example, patients with CHIP can progress with additional hits of mutations that can cause them to progress to MDS. Now, all that said, the chances of an individual with CHIP progressing to MDS is about 0.5 to 1% per year. And so CHIP is characterized by having these myeloid-associated mutations, but it's not a huge chance that they're going to necessarily progress, but there is that chance that they could. So what about people that have cytopenias and clonality? So they have ICUS, but then they also, I guess, have CHIP. So, oops, sorry. For these patients, the term CCUS, or clonal cytopenia of undetermined significance, was adopted around 2015. Simultaneously, these papers were released in Blood. But the top paper, the Kwok paper, I believe, is the one that sort of first used the term CCUS. And so essentially, in summary of all of these pre-malignant states, we've got, compared to MDS, we've got no dysplasia across the ICUS and CHIP spectrum. Occasionally, CCUS patients might have dysplasia, but like I said, it could be sub-threshold for MDS diagnostic criteria. We've got no clonality in ICUS, but clonality present in CHIP, and then the converse for cytopenias, whereas CCUS, similar to MDS, is going to have both of these categories. You can see that CCUS is looking a bit more similar to MDS. Oops. Oh, there we go. Sorry. And so I have these here as pre-malignant states, but the revised WHO classification that they're working on, the next edition, actually is going to include a subcategory that includes CCUS and CHIP, and they're going to be considered myeloid precursor lesions. So that's the terminology that's being adopted to include those, which is really important that they're being recognized in the actual WHO diagnostic criteria. So as I mentioned before, the chances of progressing from CHIP to MDS is not super high year to year. However, the chances of progressing from CCUS to MDS is over 80% by 5 years. So I don't have an exact number for year to year, but in comparison, CCUS, like I said, 80% chance over 5 years, and I think it's over 90% for 10 years. So as you can see, they're a lot closer in similarity as compared to CHIP and MDS, as you saw sort of on that chart as well. So what we're really wondering is how similar are CCUS and MDS really, and what is driving that disease evolution? So as I mentioned, CCUS has a significantly higher chance of evolution. However, that mechanism driving evolution from CHIP or CCUS to MDS or ICUS, in fact, remain unclear. So we sought to determine whether serial sequencing across the ICUS, CCUS, MDS spectrum would reveal genetic or clinical features that might be associated with disease. And this is sort of in following a group from 2017 that identified that MDS and CCUS actually had no significant difference in survival. So we're really curious sort of what is making these different, you know? Okay. So we have a cohort of CCUS MDS cohort that we accumulated with Sunnybrook. And so this includes 193 unique patients separate. This is a retrospective cohort separate from our myeloid cohort, our myeloid malignancy cohort. And so there were 240 unique samples, and that's because we have serial sequencing for a good amount of these patients, and I'll get into that afterwards. So in terms of the categories that these patients fell into, we had a few that were considered control. We had some ICUS and CCUS, as well as MDS patients, and we further subdivided the MDS patients into low risk, intermediate risk, and high risk. And this is based on the revised International Prognostic Scoring System released in 2012 that takes into account blast percentage, cytopenias, as well as karyotype abnormalities to classify these diseases by risk. And then we've got some with MDS/MPN, so sort of on the more proliferative spectrum of MDS. So we performed NGS for all samples in the cohort, similar to how I previously described for the myeloid malignant cohort. And so what we saw was that the cell counts in the CCUS patients really mirrored those of the individuals with low risk and intermediate risk MDS. So as you can see, there's a significant difference in the hemoglobin levels between CCUS and high risk MDS, as well as platelet counts, but there was no significant difference between CCUS and low risk or intermediate risk MDS. In addition to the similarity in cell counts, we saw that there was no significant difference in overall survival between CCUS and low-risk MDS. So you can see that here. We can see the significant difference between the significantly poorer survival in the MDS high-risk group, as well as the MDS/MPN compared with CCUS, but that difference was not maintained for this low-risk and intermediate risk groups. And this is adjusted for age and sex as well. So what we've seen here then is that CCUS and low risk MDS are inherently similar, sharing clinical features. But what about their clonal features? So what we saw when we looked at their clonal features in terms of the total number of variants per patient, as well as their variant allele frequencies of the variants identified, we saw that they exhibit very similar clonal features as well. So we saw that there was no significant difference in the total number of variants between CCUS and low risk or intermediate risk MDS, while there was a difference between CCUS significantly higher number of variants in high risk MDS and MDS/MPN compared to CCUS. And we also saw that there was a significantly higher variant allele frequency seen in patients with intermediate risk MDS and MDS/MPN compared to CCUS, while that difference was not seen in the patients with low risk MDS, excuse me. And so while we were working on this, there was a letter to the editor that was released looking at specifically similarity between CCUS and low risk MDS. But there's a sort of early stage and they don't have the kind of comparison with the higher risk MDS. So we're hoping to get this submitted so that we can share that information as well. So sort of the concluding part for that aspect is that CCUS and low risk MDS are inherently similar, sharing clinical and clonal features, not just clinical features. So the remaining question that we were wondering about is what is driving that evolution? So we wanted to look, as I mentioned, at sequencing across that ICUS to CCUS to MDS spectrum. So we wanted to look at serial sequencing here. So as you can see in the flow chart, of our 193 unique patients, 124 of them were sort of snapshot pictures. So they didn't have serial information. It was just based on sequencing at that time point. So some of those are our MDS high risk or MDS/MPN for comparison, some controls. We had 28 of them where we actually performed serial sequencing. So we had 2 or more samples available to sequence at different time points, up to 6 for a couple of patients, up to 6 time points, I mean. And then 46 of them had follow-up bone marrow information available. So they had information that we didn't have samples available for different time points, but we had information about whether or not they progressed down the road. And so out of these individuals here, 33 of them were qualified as, or 45% were qualified as having CCUS, and then another half or so as ICUS. And from there, we saw 33% of the CCUS individuals went on to get MDS, 61% remained at CCUS over time. And then a couple of them went on to have non-myeloid malignancy neoplasms. And then ICUS, we had some that stayed ICUS, a good chunk there, and then a few that went on to get MDS, a couple that went on to get AML, and then a couple others as well. So we saw no conclusive pattern of evolution from ICUS or CCUS to MDS, but when we subdivided these genes here into functional categories to try and increase our sort of numbers for comparison, what we saw was that there was a significantly higher number of splicing and transcription factor mutations in patients with MDS compared to CCUS. So again, not a conclusive pattern, but something interesting and potentially worth further exploring in the future. So we also saw that patients with CCUS had a higher probability of evolution to MDS early on compared to those with ICUS, which is consistent with what was shown in the Malcavati paper that I previously mentioned back in 2017. But we saw that beyond 5 years, that significance dropped off. I'm sorry that those are a little bit off. And so we're not too sure exactly why that might be. It might have to do with us having patients with MDS later sort of selected. So selection bias or sort of random chance, our ICUS cohort tended to have higher cytopenias than typically seen in ICUS. So we think that might have contributed to that difference there. So overall, for our sort of precursor lesion section, we saw that NGS is an important tool for detecting clonality in cases of idiopathic cytopenias, because otherwise we would not be reaching this diagnosis of CCUS that evidently has a higher chance of progressing and is very inherently similar to low-risk MDS. So this wouldn't be detected without the use of NGS. And so although we saw no conclusive pattern of disease evolution, we're saying that future studies are warranted to better elucidate the mechanism that might be driving that progression across the spectrum from CCUS to MDS and potentially onto AML. I don't think I mentioned before, but MDS has about a 40% chance, individuals with MDS have about a 40% chance of progressing to AML, which is a much more acute and severe disease. So catching that earlier on and starting treatment as soon as possible is also important, very important as well. So I see I'm at about 12:40. I have a case report that I was sort of holding on to if I had time. Is it all right if I keep going, or should I wrap up?
Yeah, go for it.
Okay, thanks. Okay, so this is my bonus objective. So we have a case report for a single patient that was very interesting. This patient has serial sequencing. They're an MDS patient that spans 18 years. And so how this happened, PMDX50 is their unique identifier. So they're a 74-year-old male in 2018 that had a possible MDS, but subthreshold non-diagnostic since 2003. And sort of still not fully clear picture of whether that patient was sort of qualifying for MDS, but then they started being treated for MDS in 2018. So at that time point, so this is September 2018, they had sequencing done. So this is sort of part of our cohort. And we saw the following variants that were present. But what we actually saw was that there was an archived bone marrow sample from back in 2002 from sort of this earlier on assessment. So we were able to get that archived sample and actually extract the DNA from it. And so when we did that, what we saw was that this U2AF1 S34F variant, which is a splicing factor similar to SF3B1, was actually present all the way back in 2002. So interesting that that was present there. And then this patient, we had sequencing completed again in 2019, 2 time points. And then there was also a sample from 2020 that we were able to sequence. So based on this information, we were able to sort of look at the clonal picture and see how the ETV6 and PRPFA variants here, you see that they start to decrease in variant allele frequency over time. And they kind of vanish altogether, while the SETBP1 and the other U2AF1 variant are increasing. And so you can see sort of here a picture where this variant, clone, we suspect goes on to not be detected, outcompeted by other clones. So I have sort of a better picture of that. So what we're seeing is, this clone was the predominant one in this U2AF1 harboring clone, acquiring additional mutations, starting to take over and drive that disease up to the point where they were getting sequenced in 2018. But progression where additional variants were acquired in sort of an original clone, that original clone here, where they're sort of outcompeting each other. And then this clone became the predominant clone that's got some of these other mutations, the RAS mutations. And then this patient subsequently, unfortunately, expired in late 2020. So just a very interesting case looking at how serial sequencing can really help inform sort of the disease progression for patients. And then I've got one more. Oh, we were also curious, based on this here, how you can see this is around the same VAF. So I was asked this a while back at a seminar when I presented this case. So how do we know that this is not a germline variant, right? Because why is the variant allele frequency remaining so consistent over time? So we actually wanted to confirm that this variant was a somatic variant. So we were able to obtain an archived FFP colon tissue from this patient that was previously collected for an unrelated reason. And we received ethics approval to extract the DNA and perform sequencing. And we actually saw that that was present in about 7% in the tissue, which is consistent with leukocyte infiltration into the systemic tissue. So we were able to actually conclude that it is a somatic variant, given that it was not present around that 50% VAF. So this was also interesting point for another, I'm not going to go into too much detail about this, but there was another paper that had postulated that the S34F and Q157 hotspot variants, which this patient also had, that second U2F1, that they occurred around the same time, but we have evidence here showing that they're occurring sequentially. So we have a case report looking at that as well. But so more importantly, in the context of what I've been talking about the rest of the talk today, is the importance of detecting these variants early on. So what we were able to do, because this patient had sequencing across so many time points, is go back and look in IGV, or integrated genomics viewer, at the individual positions where these variants occurred later on at a time point, to go look at the earlier time points and see if they might have been present at a level below the clinical reporting cutoff or ion reporter sensitivity cutoffs. So what we actually saw, for example, is this KRAS variant, for example, was present at a level of 0.28 back in 2018, when the patient was sort of finally confirmed to have MDS, as was this ETV6 and these additional mutations here, but none of these were called. You can see sort of the plus in the initial sequencing because of their low variant allele frequencies. And then there's similarly some over here, for example. And then what we also saw is the importance of having, comprehensive variant analysis workflows. So the Oncomine filter is the filter that was initially released with the commercial Oncomine myeloid panel. And so the variants that were identified with that filter are included here, but then there was a more recent version released, the Oncomine extended filter. And so you can see the ones with sort of this double cross are the ones that were not picked up by the Oncomine filter, but actually detected with the Oncomine extended. So, really showing the importance of how this clonal picture changes for this patient if we're not detecting all of the mutations that we could be detecting, and whether, for example, they would have started their azacitidine treatment earlier on, because they didn't start that until 2019, and when they were a bit higher risk, but perhaps that would have been started right away at 2018, for example, when they were diagnosed. So overall, what we've seen today that variants detected in myeloid disease are clinically actionable, and that CCUS and low-risk MDS are really inherently similar, therefore making it very important that NGS be used to detect clonality and idiopathic cytopenias, to be able to detect CCUS, modify follow-up for those patients, and then maybe potentially this and other research will sort of change that concrete arbitrary cutoff that qualifies a patient as low risk MDS. Maybe that bar will be lowered a bit to include some of those CCUS patients, considering how similar they are to those low risk MDS patients. I also demonstrated with that case report the value of serial sequencing and comprehensive variant analysis workflows. I have another short paper about the variant analysis workflows, but for the sake of time, I figured we wouldn't get into that today. And so one of the future directions, especially out of the CCUS side of things, is exploring other sequencing modalities to better elucidate that mechanism that could be underlying the evolution from CHIP and or CCUS to myeloid neoplasm. So things like single cell sequencing and error-corrected sequencing, RNA-seq, to see if any of those will help better demonstrate what might be driving that evolution. So we're hopeful that that the work that we've disseminated and that will continue to hopefully get published will demonstrate the importance of sequencing for myeloid malignancies in general, not just AML, across the MDS, MPN, MDS/MPN spectrum. And also, I wanted to just sort of reiterate what I was saying before about how these myeloid precursor lesions are set to be included in the newest revision of the WHO guidelines. So just demonstrating that research is starting to be considered, and that these are starting to be included in the diagnostic information for these patients. So I'd like to thank everybody that's contributed to this work, my supervisor, Dr. Michael Raw, and everyone from the Raw Lab, as well as my supervisory and PhD examining committee members, and our collaborators at KHSC, Sunnybrook, and St. John Regional Hospital, as well as within the department. And of course, our funding agencies and CIHR for the CGSD award, as Dr. Cody mentioned at the beginning. And thank you all for listening. This is just a couple of the wonderful opportunities I've had to attend conferences throughout my degree. On the left, we're in Vancouver, some lab members, as well as in San Diego at ASH in 2018. And this is in Copenhagen at the MDS International Symposium. And then in the middle, you can see the bone marrow that Mike thought that we should all try, considering our topic of research. So we all tried that in Vancouver, and it was pretty good, actually. Thank you. Happy to take any questions.
It was a fantastic seminar, Christina.
Thank you.
I really enjoyed it, and it clearly shows the path towards personalized precision medicine. It's very exciting work, and I saw the reminder popping up for lab meetings.
Oh, sorry, I didn't realize
but
that
since
was on
Michael
the
is
screen
here,
there.
and I'm sure your lab members are here as
Yeah.
well, you can
Yeah. No,
consider this
sorry.
as a
It
lab.
Continue.
pops